several. We have three classes of the cyclins. So G1 cyclins, they're responsible to moving the cell through G1 to the S phase, and the S phase cyclin initiates and maintain the DNA replication, etc. The three kinds of the kinase. And what about the cyclin depends the kinase? So they are master regulator of the cell cycles, and of course they're working together with the cyclins, and they are kinase. It means that they are enzyme that needs to attach one phosphate group into them. And they active only complexes with the cyclin proteins. So here is a picture about the CDK2. A cyclin binding to this CDKs will cause the conformation change in the CDKs. Here's when they are attached with the cyclin A, the conformations change, the telos change positions, and also the structure substrates will be assessed. So you can see here they are in closed images, they in the closed states. And when one cyclin come with them, they will open, and the subjects will come here, and they will work. And the cyclins depend on kinase. They are really evolutionary concept. And for the yeast cell have just only one CDKs, but in humans we have a multiple CDKs. You're in different states of the cell cycles. Like we have a CDK4 and 2 in the G1 state, and they will connect with the cyclin D, and G2 are connect with the cyclin E. And we have a CDK2 complex with this cyclin A. Is exactly the example here. So why we can discover about the cyclin dependence kinase is maybe in the same case we discovered the cyclin. That's this graph illustrates the different tracer of the single CDK1's activity stimulates different events during the fission of the yeast cell cycle. So they also found that in the mitosis, numbers of the CDK1 and the cyclin B activity, they increase in the metaphase. So different level of CDK cyclin activities requires for two transitions. A lower level is promoting S phase, and the high level promoting in mitosis. Low level here and high level here. We have also one terms that's maturation promoting factors. And they are famous example how cyclins and the CDKs work together to control the cell or cycles. Here the maturation promoting factors, you can see they are increased during the mitosis. But it's just an examples of the CDK1 and the cyclin. So I don't need you to remember these slides. So CDKs and cyclin complexes they regulated in several ways. They could be regulated by the phosphorylations. Like here's when the CDK1s, so in the first stage, phosphorylation, they combine with each other in the active form, and when one phosphate group here, actually they want ATP comes and leave one phosphate group in this complex, they will be active. So the cell will be moved from the G2 to the mitosis stage. And Another case, proteolysis here. They are in active form between CDK1 and cyclin B. The mitosis, when they want the mitosis, and sorry, it's not really like this. When they want these are and the mitosis and moved into the Z1 stage, they could be one enzymes go to degrade the cyclin B. So it means the complex CDK1 and cyclin B separate now and they in the inactive form. And another type that is the inhibitor, the Euro inhibitor. That's one kind of proteins or enzymes can bind into one CDKs in active form, and they will become inactive. Or one kind of inhibitor bind here in by rise or there's in the this active complex, and they can become inactive form. So and the another regulations process, it could be the subcellular localizations, like during the G2, the NES. Remember NES? One kind of tail that have some things export out of nucleus, remember? So during the G2, the NES is mediated nuclear export of cyclin B dominates, resulting in the separations of the cyclin B and the CDK1s. And as mitotic commitment, phosphorylation of the key residue in our cyclin B. So here in the mitotic commitments, so the cyclin B attenuates export, resulting its cells stays in the nucleus. So they separate cyclin B separates or they in the nucleus, so they cannot meet the CDKs in the cytoplasm. Okay, so they separate from each others. That's one kind of subcellular localization. For this pass, I will skip for you. Cell may withdraw from the cell cycle to go to G0 stage, or I tell you before, okay? And also it's about the checkpoint. You know that the cell is